morning students. Today we are going to start the next section of the enzyme chapter, that is cofactors. Cofactors means what? They are the non-protein part of the enzyme. And what is the significance? It's necessary for the enzyme function. Different types of cofactors are there. There are inorganic cofactors and organic cofactors. Inorganic cofactors means metal ion cofactors are inorganic cofactors. Then we need to understand the mode of interaction of that metals in enzyme. How that interaction, how that metal present in that enzyme interact with the enzyme. Metalloenzymes are there. Where the cursor? Metalloenzymes are there. In this case, metals are tightly bound to the enzyme by coordinate bonds. Example, iron. It is required for cytochrome oxidase catalase. That is metalloenzyme. Then metal-dependent enzyme. Metal-dependent enzyme means in this case that metal is loosely associated with the enzyme, but its presence is necessary for the catalytic function. Example is magnesium. Then metal associated enzyme, sorry, not associated, metal activated enzymes. It's a mistake. Not associated, activated. Metal activated enzyme means these enzymes get activated only in the presence of metals. Example is chlorine. Then another type of cofactor, that is organic cofactors. Organic cofactors are classified based on the nature of binding with the enzyme. How it bind with the enzyme. Organic cofactors are of two types: prosthetic group and coenzymes. Prosthetic group and coenzyme. Prosthetic group means that cofactor binds very tightly through covalent bond to the enzyme. And after the catalase reaction, it can return to the original state also. Anyway, the important thing we have to understand is that prosthetic group means that cofactor which is tightly bonded to the enzyme. The coenzymes means it is non-covalently and loosely attached to the enzymes. They undergo certain changes during the reaction. And that prosthetic group will not undergo changes during the reaction. Then we are going to learn more about the coenzymes. Coenzymes, major coenzymes are of two types. For that, we need to understand some more thing about coenzymes. They are heat stable non-protein part of enzyme. Coenzymes are. It is stable in heat stable non-protein part. It also loosely bound to enzyme, and sometimes it form a part of active site. And since it behave like a substrate called substrate, it is called as co-substrate or second substrates. These are some additional points we need to understand about coenzymes. Then different types of two types of coenzymes are present. Coenzymes involved in oxidation reduction reaction, and they are called NAD. And the examples are NAD, NADP, FMN, FAD, C enzyme Q, coenzyme Q. Then next type is called coenzyme involved in transferring groups other than hydrogen or electrons. They are involved in reactions takes place through transferring certain groups instead of the transport of protons or electrons. Examples are ATP, ADP. Then we are going to see one by one about this coenzyme. First one is NAD and NADP. What is NAD or NADP? They are coenzyme forms of B complex vitamin. B complex vitamin, that is niacin or nicotinic acid. It is the coenzyme form of B complex vitamin niacin or nicotinic acid. What is about this composition? It consists of a nucleoside adenosine linked to a modified form of niacin. while we are going to see that structure of the NAD, it will be more clear to you. Now you need to understand in this way. Composition, it consists of nucleoside adenosine nucleoside. Adenosine, when thymine, uracil, so many nucleosides are there. Similarly, adenosine nucleoside, it is linked to um, a modified niacin group that forms that NAD. Then what about NADP? NADP means there nucleoside adenosine is there, niacin is there, and in addition to that, a phosphate group also will be there. That is NADP. Then some popular name also there for this NAD and NADP. NAD is called coenzyme 1, NADP is called coenzyme 2. Then we are going to see the mode of action. It is mentioned that it involved in oxidation reduction reaction. As it is involved in oxidation reduction reaction, they are carriers of protons or H plus. And after the reaction, it can detach from the enzyme part also. So its representation is given here. SH plus NAD plus gives P plus NADH. This explains the condition which substrate is undergoing oxidation. Substrate is yes. Now the substrate is having that H proton. It is going to undergo oxidation. Oxidation means taking off H plus or electrons. That H is given to this NAD. That means NAD is get reduced. So that's why product is forming along with the NADH. That way NAD is involved in oxidation reaction of this substrate. And reduction, how it involved in reduction NADH. Here in this equation, substrate is in oxidized form and NADH is the reduced form. And here that NADH is giving this H to this substrate while forming the product. So this NADH undergoes oxidation and the substrate undergoes reduction. Okay, that's why this NAD and NADP are cofactors that involved in oxidation reduction reaction. This is the structure. For some examinations, university examination, there is questions like as the students to draw the structure of NAD and NADH. So it's very important to learn how to draw these structures. For drawing the structures, very easy. This is adenosine. Adenosine. This orange mark. This is the sugar. This is the phosphate. So, this is the nucleoside. This is the nucleoside. Sugar is there. Adenosine is there. To this, what is adding? Niacin is attached. When niacin is attached to adenosine nucleotide, it become NAD plus. When it become NADH, an additional H plus is coming here. Then it become NADH. Next is flavin nucleotide coenzyme. What is flavin nucleotide coenzyme? Coenzymes derived from vitamin B2 or riboflavin are called flavin nucleotide coenzyme. It also exists in two forms, FMN and FAD. FMN, where is it? FMN and FAD. FMN means flavin mononucleotide. FAD means, where is it? FAD means flavin adenine dinucleotide. This FMN and FAD, FMN consists of phosphate group bounded to riboflavin. FMN means it consists of phosphate group bonded to riboflavin. Then what is FAD? It is formed by linkage of AMP to FMN. So look to that picture. This is the riboflavin. Riboflavin means vitamin B2. To this vitamin B2, when phosphate group attach, it becomes flavin mononucleotide. That means FMN. Along with this flavin mononucleotide, when an additional AMP unit attaches, then it becomes flavin adenine dinucleotide. For this one also, you should learn how to draw its structures. Can come as questions for the examination. Mode of action, how it is also involved in oxidation reduction reaction. FMN plus and FAD plus are the oxidized state, and FMN H2 and FAD H2 are reduced state. Wherever plus is found, it is the oxidized state. That is what you need to understand. Next factor is coenzyme Q. Coenzyme Q, its chemical name is ubiquinone. It is a lipid material and it is abundant in mitochondria. Okay, for biological oxidation, we are familiar with that cofactor. And it is involved in oxidation reaction, and it can remain oxidized as well as reduced state. Next cofactor is adenosine nucleotide. It is a group of 3 coenzymes, adenosine nucleotides. Means that group means AMP, ADP and ATP. Adenosine monophosphate, adenosine diphosphate and adenosine triphosphate. And what is its role? It is involved in transphosphorylation reaction. It involves transferring which group? Transfer of phosphate group. So that's why it is involved in transphosphorylation reaction. How it is formed, this adenosine nucleotide? Through phosphorylation of adenosine. When adenosine get phosphorylated means when two adenosine phosphate groups are added, then forms AMP. If one phosphate group is added, it become AMP. Two phosphate groups are added, become ADP. If three phosphates are added to adenosine, become ATP. They are called the energy currency of cell. Then next cofactor is pyridoxal phosphate. Pyridoxal phosphate is actually derived from a B complex vitamin pyridoxine, that is B6. From B6 vitamin it actually derived, and it, as I said, it acts in transamination and decarboxylation reaction that you all learned in the former chapter. Then thiamine pyrophosphate. Thiamine pyrophosphate, it's a derivative of vitamin B1, thiamine vitamin. Its another name is co-carboxylase. It causes the carbon dioxide removal from alpha keto acid. What it do? It remove carbon dioxide from where? Alpha keto acids. The important reaction that is involved by this thiamine pyrophosphate is decarboxylation of pyruvic acid to acetaldehyde. In that reaction, this cofactor helps significantly. Then coenzyme A. They are molecules with the free sulfhydryl SH groups. These are cofactors with the free sulfhydryl groups. It also involved in acetylation reaction, that is transfer of acetyl group to some other substrate. Vitamin pantothenic acid is the major component of this coenzyme A, which is vitamin pantothenic acid. By this way, we finish this chapter and today's section. Thank you.